Okay, I'll get started today anyway. So where are we? Sharing my screen. Let me just get back to the main page. We're on our main page, rockefelleruniversity.github.io. I have the videos. I'll put up last Friday's video after this meeting, and I'll try and put up our video from today. Covered our introduction to R, covered plotting. We covered a little bit about types of data we'll be handling, genomics data, how we can review it in IGV. And then we had, hey David, we have started our introduction to Bioconductor, which really is just covering how we will handle the objects which we're going to need to use for RNA-seq, ChIP-seq, ATAC-seq, and single cell analysis. So we did our introduction. We had a little look at how we handle sequences in Bioconductor, and primarily this was about FASTA files, but we're going to come across the same objects to handle sequence data today. We looked a little bit about genomic intervals, how we can store start-end regions in the genome. We looked at genomic scores. So this is not just start-end, it's start-end with a score. So that can be a number of reads aligning to that region, conservation of that region across, say, different genomes, different species, GC content. And then we looked at genomic features, which is what handles our GFF, GTF objects. And it's these we will use for counting over gene models, or even, as we'll see later on, annotating peaks to their nearest TSS, peaks to the region of a gene. Great, so now we're going to start covering actually directly some of the data we'll generate from high throughput sequencing, and we're going to start with one of the most basic or earliest files you'll see coming back from a genome center, and that is FastQ. Okay, so there's a little bit in the lecture today. I'm going to step out from the slides and show you one of our pieces of software just because it's useful, but it's still important to know how we handle Bioconductor in Bioconductor sequence information, so this FastQ data. Okay, so how much time have I got? 5 minutes in. Both sessions today should be quite short, but please feel free to ask any questions. So we can now massively parallel sequence DNA fragments using Illumina Sequencing sequencing. I don't think it's called Sequencing anywhere anymore. Just remind us, Sequencing was the original company. Illumina, I think, bought them and became the Illumina we know nowadays. This allows us to rapidly profile abundance of specific DNA sequences in a pool of DNA sequences. So as long as we can make DNA fragments, be it from fragmenting our DNA, right, our actual genome, or in fact, making copy DNA, which is just a copy of the RNA, we can then sequence this and get an idea about the sequences in our pool of fragments. Because now we have the full genome sequence for our major organisms, or even less major organisms, we can actually use the full sequence to align, map our DNA fragments from our data to the full genome. And then we can get an idea where our sequences are mapping. That's kind of useful. Whether the sequence in our sequence differs from the main genome, or even in combination with transcriptome models, we can actually estimate the abundance of transcripts genes by, say, capturing the cDNA, sequencing it, mapping it to our full genome, seeing which land inside transcripts or genes, and using that as a proxy for abundance expression. So sometime in RNA sequencing, we'll get Connie, who is our genome center manager, to give a better explanation of Illumina sequencing, but let me try anyway. So really what we do is we start with our fragmented DNA, in this case, double-stranded DNA. We'll add adapters to this, right? So adapters which allow us to do Illumina sequencing. This then gets made into single strand, and the Illumina flow cell, which is this object we see here. It's sticky and it's got primers or complementary bases here, which will then allow these DNA fragments to become attached to this flow cell. So this is the flow cell surface here. So we've got our double-stranded DNA, we add some adapters, and it's these adapters which allow the DNA then to stick to the flow cell. We can then do something called bridge amplification, and we will just amplify, similar to PCR, we will just amplify the fragments we have on our flow cell until eventually we will have a collection of fragments on our flow cell which are all identical. We take our DNA, we stick it down to our flow cell by using these adapters. We just copy and amplify this DNA until we have what is called clusters of DNA fragments attached to the cell surface. And these clusters will all have copies of the same DNA fragment. So then it actually comes to sequencing this. What we do is we add in cycles different bases. So we will add, say, there are 4 different bases, and each one of these will be colored. So they have a fluorescence to them. And I'm not sure if this is actually the colors, but say guanosine is yellow, C is blue, A is red, thymine is green. So every time we add these colors, we have a cycle, and one base gets added to our DNA fragment copy. So we're kind of copying, making a copy of these clusters by adding one base at a time. And we can actually photograph it as we add this one base. And we can see, okay, in this cluster, in order to make a copy, we've added a G, so we get an idea of the sequence for that cluster. So we're imaging this flow cell, and we're saying, okay, we see a green here, which means we've added a T for this cycle of sequencing, and then we'll stop, we'll wash off everything, we'll add the bases again, and maybe at the same point, we'll now see a G, and that means the next base in the sequence was a G, and we just continue doing this until we reach the end of our sequence. So in one spot, we may have our DNA we want to sequence, and we have an A, a G, third cycle is a C, fourth cycle is a C, and over 100 cycles, we'll get 100 base pairs sequence for every single cluster. Okay. At the end result, we have usually a program. I think in Illumina, it's BCL2 FastQ. You often never see this. This will be run by your genome center. And effectively what that does is turn these stacks of images into sequences. Okay, so given the colors fluorescing in the different cycles, I can then say, oh, I have a sequence of AC, AG, CC for this particular cluster, and that will be one of my reads. Illumina sequencing now, you're going to get up to 1 billion reads on a flow cell, whereas originally we were only doing, say, 8 million with the early ones, maybe 2 million. So we can really pack a lot of clusters now onto these flow cells. Actually, the accuracy, so how well we know that this is really red, that's what tells us how well we've sequenced a particular base. So if this red, we weren't quite sure if it's really red, maybe it's a little greenish. That quality score, this quality scores we see here, that's what really that is based on. Okay. How well we can really recognize the color for that particular sequence, that particular cluster. So we saw earlier on, the product of this whole process is a FastQ file, right? And if we look, hopefully maybe we're going to come back to it, but we recognize some components of this. We have our read name, which is our kind of actual machine there. And then we have here the position on the flow cell. So that's the actual position of this cluster. Okay, so we know where this cluster was in the flow cell based on this position. And then we have the sequence, which is the construction of all these colors. So ACGG, or AGCC, sorry. So do that 100 times, we'll get a sequence. And then we got our plus line. And then these are our Fred quality scores, which is our encoded quality scores. Effectively, we use a character, an ASCII character, to tell us, what bin of quality this base, the corresponding base, really is. So a low quality base, such as these hashes we saw earlier on when we looked it up, probably comes from a place on the flow cell where we haven't confidently called that base to be an A, a C, or a G. So Illumina sequences as FastQ files can be handled in Biocondutor using a package we've already seen, biostrings. So we already used the biostrings package to handle faster files. So we were using BS genome packages to retrieve it from the database, but then the actual biostrings package is what allows us to handle FastQ files. And actually it's the ShortRead package, because we're handling ShortRead data, which allows us to read in FastQs, manipulate them, and do some actions on them. So to install any package, we know that we just need to go to the Biocondutor page. So if we Google that, it'll take us to Biocondutor. I can just copy and paste this into my present R session, and it will go and install that package. Okay. So we're going to make use of some encode data if you want to follow along today. So this is raw sequence data. It's about as raw data as you'll find. Generated from an RNA-seq experiment. It's actually generated from a human cell line, GM12878. You can find the link here, or if you just want to download and follow along, you click that and it will download it to you. So to make life a little easier for us, but you can apply these things to any data, but just so that you really can follow along, and so I can compile it on my computer. I have sampled the first 100,000 reads. From this file. And then this should allow us to quickly perform some of the steps. So we've sampled it and we've put it in the data directory. The original FastQ, as you download from here, it's a little bit bigger. Okay, so I'm going to show you I'm going to show you a little bit about how we can handle really big FastQs later on in the session. Okay. So most of this class, most of what we teach you, in fact, all of what we teach you, you can really apply to your own data, gigabyte data, but just for the class, we're going to use a smaller data set. But at the end of the session, I'll show you how you can save memory and loop. So to read in a FastQ file, it's super easy. We just load the ShortRead package, library ShortRead, and we can do our read FastQ function. I think that just takes one argument here, and that's the path to our FastQ file. Doesn't matter if the FastQ is zipped, so often you'll get FastQ as gzipped. Most read programs in R can handle gzip files. You don't need to tell it it's zipped. It'll just read it in. So once we've done that, we can do read FastQ, and we get back an object. This is a new object class for us. We haven't seen this before. It is a ShortRead Q object from the ShortRead package. Okay, so we don't know how to deal with a ShortRead Q object yet, but if we type in the object to the screen, it tells us not much information, but it tells us the class is ShortRead Q. And it tells us we have 100,000 reads and we have 76 cycles. So remembering cycles is every time it adds a new set of DNA and it sequences another base. Right, so every time it adds these colored DNAs and it sequences another base, that is a cycle. So when it says 76 cycles, it actually means 76 base pairs long. We can use our familiar length functions. So a lot of the standard functions will work on this object. If we want to get the number of reads, we can just do length FastQ, and it tells us we have 100,000 reads. We can use a standard vector length on this object. Sorry. We can also use the width function. So similar to how we use the width function on G-ranges or DNA string sets, if we use the width function on our FastQ object, it will give us back the length, the number of bases in every single. So right now, everything's 76. When you get sequencing straight from your genomic center, you will expect them all to be the same length. Sometimes you will pick up sequences from other people, and you might find that actually they've done trimming or filtering of the data, and your reads are no longer all the same. We can subset or index our short-read Q objects using the same methods as we have for vectors. So if I want to look at just the first 10 reads, I can just do FastQ, and I can provide an index for the first 10 reads. So 1 to 10, this is going to give me the first 10 reads. Any indexing, which we can use for vectors or lists, we can use here. And you can see I get back the same class object. So I just have 10 reads. Obviously the same length. I haven't filtered or trimmed my reads yet. So FastQ data, if we type it in, we can index our object, make it shorter. But we're not really sure how to deal with this object yet. It does tell us the maximum read length. It tells us the total number of reads. But if we want to get into the actual object itself, we can have to use these accessor functions, SRead, so sequence read, which will retrieve the sequence of the reads, quality, which will retrieve the quality of the reads as ASCII encoded scores, and IDs, which will retrieve the IDs of the reads. So we're going to be able to grab the 3 elements we really need. This SRead will grab us the sequence. IDs will grab us this ID here. And quals is going to grab the quality line. So we have ways of accessing this now. First thing we can do then is get the read sequence out of our FastQs. So we can use the SRead function here, just apply it to our FastQ object, and we'll get the new object back called, we're naming it here sequence of reads. But if I look at the class, we can see it's a biostrings package and it's a DNA string set object. So the SRead is bringing out the sequences from our FastQ object, but now we have something we should recognize, a DNA string set. DNA string sets are what we were using previously to store FastQs. Right? So just to remind ourselves, a DNA string set is just a slightly better way of holding sequence data in R. If you look at the sequence of reads we just generated, it just shows us the first 5 and the last 5 from our 100,000. So we can see like lots of the sequences are N, so it's poorly sequenced probably. We have a few bases here. And then we have basically a record for every read in our data set, and we have the length width here. So we should know how to handle these, hopefully, but not. We can look at some of the most important things we can do with this sequence in a little bit here. So hopefully one of the things we remember is alphabet frequency. We did this when we wanted to. It's one of our exercises as well. So alphabet frequency is from the biostrings package, and it does this in a very fast manner. So we'll go through our reads, and it will give us the occurrence of each base across the reads. So here, I'll do alphabet frequency on our sequence of reads, and if I look just at the first 2 now, okay, because we can see we have a matrix, and we have column names by the base, rows are the reads, and for the first one, it's saying we have 68 N's, 2 A's, 3 G's, sorry, 3 C's, 1 G, 2 T's. It's hard to tell really, but we can definitely see 2 T's, lots of N's, 2 C's, a G, and there's our 2 A's. The nice thing about this is that, if we're worried about bias in our data, we can use alphabet frequency and we can start to see, do we see more G's and C's in our reads than we would expect by chance? Do we see some reads which are, either mostly unknown, or do we see some reads which are biased to having A's at the beginning or end? And these are the types of things we can now do now that we've extracted this alphabet frequency. We can also extract the IDs for our sequences using ID. So this is going to give us another biostrings object. So this is a slightly stranger object. It's a BS string set. So there's no DNA associated to this. It doesn't think it's DNA or amino acid. It's a much more generic object. But we can look at it and you can see that it's very similar to our previous the biostring sets objects. We have our read name here, and we have our width. So it's this type of thing you'd want to parse, and you'd probably parse these last things here if you wanted to start to say IDs in a certain location on my FastQ flow cell, my Illumina flow cell. Had a problem. This is, I'm not quite sure the setup, but effectively this is your location on the flow cell. And we can do everything with the BS string set that we did with DNA string sets. We can convert them to a character if we want to. If we're more comfortable working with characters to start to parse this, we can just do as.character, give it the IDs, and then we'll have our 4 IDs. Maybe that's just easier for us to work with. Now the major difference between sorry, having a bit of a issue with the projector. There we go. I don't know what to do really at this point. I'm going to change over one more thing. Might be you just have to struggle with the yellowness for a little bit. It's the projector, right? Not me. Okay, we're back. Oh shit. Oh, well. Sorry. Would you rather I turned it off, or would you? My answer as well. I'm not sure what's good. I'll leave it for now. See if it becomes too much of a pain. Sorry, I got to find my notes again. Okay, lovely. So a fundamental difference between faster and FastQ is the quality score. Sorry, we're having problems. Okay. Fundamental difference between FastQ and faster files is the Q for the quality score, contained obviously in FastQ. So FastQ offers a little bit more than just faster, in that it allows us to so it allows us to really tell how much we believe the sequence at a particular position in the faster file. Okay, so as we've seen before, quality scores are actually encoded as ASCII characters representing the -log10 probability of a base being wrong. So larger scores will be associated with more confident base scores. Yeah, I'm not really sure what to do about the. Yes, please. I get the feeling it's actually the adapter. I'm doing that now anyway. So I don't think it's the adapter. Yes, please. That'd be great. Okay. Sorry, guys. You'll have to just follow along the slides, I think. So wonderful, horrible blue thing going on. So for a really good description of this, we can look at FastQ. And they got a description on their page here where they talk about the encoding of FastQ qualities, like here. So ASCII qualities, how these actually relate to scores. So one thing worth noting is we have a lot of hashes in some of the data we just looked at. Actually, hash, I think, was at the end of the reads. So if we look again at our qualities, you see we have lots of hashes towards the end. So it tends to be that as you get towards the end of the read, as the reagents run out in the sequencing, tend to get lower quality towards the end. Okay, so we can actually get access to these quality scores using the quality function. Oh, I'm not sharing the screen. I'm zooming. Sorry. So we can actually extract the qualities using this quality function. So just as we had sRead to get the sequences out, IDs to get the IDs out, we have qualities. We apply that to our FastQ ShortRead object, and we can get back a BS string set object, but now a special type of this object, a FastQ quality object. So this object itself contains our quality scores as characters in ASCII format. So we just shown you, or I just said, hash tends to be a very low quality. So you can see these first reads where we had mostly ends. But we weren't sure of the actual bases. Typically, you expect those to be very low quality. You can see here we have same length, same width, but very low quality for the original sequences in this file. If we want to translate these into something more numeric, we can actually do that ourselves by first looking up the encoding from our qualities. It's going to tell us what the spread and what values are available to us. So if I just pass our quality object from here to this function encoding, you can see it returns back a named vector with the names of the encoded quality scores, and the actual numeric value associated to them is underneath here, is the actual value. Okay? So we can actually look up the encodings ourselves, and then we can see that B is actually quite a good quality base, hash is very bad. We had some B's here, so maybe those are good quality reads. We then want to make use of this. We can actually extract the quality reads from our FastQ quality object using standard vector indexing. So if I want to get a FastQ quality object for the first base, I can just do single square brackets, and it's going to send me back a one length FastQ quality object of length 1. So like similar to lists, if I do the single square bracket, I get the same type of object back. If I do double square brackets, it actually goes in to that object and just pulls out the BS string object for that entry. Okay, so this is going to give me back a shorter FastQ quality object of length 1. This will actually get me the underlying quality scores. I can then use a little trick, which is a really useful function, string split. So I will translate our qualities into a character. I will then split up my character string by, here I'm just putting an empty, and it gives me back a vector the same length as my string, but now every base is a different entry. So I have here probably 76 entries of all hashes. Okay. And I can then pass this list of hashes here, sorry, vector of hashes as names to my quality encoding object I just created here. And what it's going to do is it's going to use that values I passed it as the name. So it's going to index it and pull out the values associated here with hash. So by doing that, I'm going to pull out all the values associated with the qualities. They were all hashes. So now I can see that all my quality scores were 2. That's very good. Thankfully, we don't actually need to calculate that ourselves. That's a little bit painful. Multi-step process of getting the qualities, splitting up every, getting the encodings, splitting up the quality for every read, and then using that to translate. We actually have this function alphabet score, and we can just pass in the full qualities object, and then that's going to give back a vector here, the same length as the qualities we pushed in, but with the summed quality score for it. Okay, so already we can start to look at things like what is my overall quality of reads, and maybe start to drop some reads which had qualities very different from the, say, median, mean, or just sticking out from everything else. We can check that that is the same as what we did, just using our kind of quality encoding ourselves. Yeah, it will give the same value. It's just alphabet score will be much, much quicker. We can also pull out a matrix of quality scores over cycles for all reads by using a slightly strange looking function, but we can do this as we give it the quality scores we have, and we tell it we want it to be a matrix. And what this actually returns is a matrix of quality scores where the columns are the cycles, so the position in the read, and the rows are the different reads. Okay. Just to check that, we'll take the row sum of the first one, and we can see it's exactly the same now as we have for these two. The advantage of this is I'm doing row sums, but you could also do col sums, right? So you could also see is my first base higher quality than my last base. And that's something you'll be doing in the exercises as we get there. The ShortRead package has many functions built really for FastQ to allow us to get useful metrics. Another really useful one is alphabet by cycle. So this is going to give us a very quick way of summarizing for each position in the read. Do we see more A's, C's, G's, or T's than we may expect? Okay, so we can see now for the first 4 base, or the main 4 bases, A, C, G, T. We can look over the first 14 cycles. And although we don't really see much, maybe there is a little bit more A's at the beginning, and that slowly comes out. It is quite common, and you'll see this in the exercises, that you will have bias at the beginning of your read, right? And this is because often some high throughput sequencing techniques will chop up or fragment your DNA in a sequence specific manner. It also tends that when you sequence, tend to just sequence more A's and T's at the beginning of your sequence. And overall, we might want to see if GC bias gets worse over time. G's and C's tend to get sequenced better. So capturing this information is really important, and it's super easy with ShortRead. We can also just apply this to also our qualities. And although these qualities are still encoded in this weird alphabet, we can get an idea about how the qualities maybe change over time as well. Another really useful function is just to see if you have too many of one sequence in your file. So here we can do table, and table will identify the number of times a sequence appears in our FastQ sequence reads. So in this case, we can just do table, give it our reads, And we can see that our most common read is in fact A and then a whole sequence of ends. Okay, so you can use this to just quickly see what is your most common read. Is there some bias, something you're sequencing over and over again? You can pull that out just using the table function. If you want to, you can also remove duplicated reads from your file, and you can do this by using the SR duplicated function. So what this does is actually just returns a logical telling whether or not a read is duplicated. So you could actually use this then just to maybe take those which are duplicated or those which aren't duplicated by taking this logical and just filtering down the original ShortRead Q object. Again, there are slightly better ways of doing this, but what often you'll see a duplicated read is actually arising from PCR over-amplification. Okay, so actually that really depends on what you're sequencing, what actually you're sequencing. But if you're sequencing a whole genome and you're not going very deep, it's very unlikely that you would sequence exactly the same read twice. If you're doing ChIP-seq, it's also quite unlikely. So we like to remove duplicates from ChIP-seq. RNA-seq, there's things like actin beta. It's so well expressed, and it's actually quite short, that you do expect to see duplicates there. So, you know, removing duplicates is really dependent on your sequencing you're doing, but having a measure of your duplicates is also very important. So here, we can just do table duplicates, and we can get an idea how much of our actual total data is duplicates. RNA-seq, you expect higher. ChIP-seq and ATAC-seq, maybe you expect a lot lower. We're going to cover the very specifics when we talk about this lectures. So we have a few different ways of doing quality control. One is using our own package, and one is using the ShortRead. Okay, so actually for quality control, just quality control rather than reviewing your FastQ reads directly, I recommend our FastP is super fast, and we wrote it because this is too slow. But ShortRead does have a function called QA, which is their quality assurance. You just pass it the path to the FastQ file, and it will go and build a whole set of metrics for you. Importantly, it has this function report. You just pass it that object, and it will make a report on your computer into here, a temporary directory, and just open it for you. If you want to actually save that report, it's going to do it to a temporary directory here. You just need to provide an additional parameter, dest for destination, and tell it I want it to write it to this directory, and it's going to create a directory with index.html. Once you've got that, you can just do browse URL. If you ever want to open up any URL from inside R, just do browse URL, give it the report you want to open. So in this case, this. And it will go and open that file for you. Like I was saying, I'm not sure if we saw much evidence for it, but often you will see lower quality reads towards the end. Sorry, lower quality bases, like here, these hashes we know are low quality, towards the end of a read. And this is because you run out of reagents, the machine is starting to run out of reagents, and just tends to give lower quality sequencing. So we can actually control that, or we can clear out some low quality bases at the end of our reads by using functions like the trim tails function. So the trim tails function is going to remove reads from the 3' end, which fall below a desired quality. So it accepts the minimum number of successive bases required to be below the quality cutting cutoff, and what this actual cutoff here. So here, it's very kind of strange, but let's look up the actual help. Oh, sorry, let's make this bigger for a start. I'm installed me. How's it going, David? Did you find anyone? Yeah, yeah. We need a ticket before we come. But the boat's sinking. We'll look at what Trimtails does. So K, describe the number of failing letters required to trigger trimming. A, with intro, giving the letter at or below which a nucleotide is failed. So effectively, in this case, we need, this is our minimum quality score, and we need 5 bases in a row to fail from the trim tail's end. Just, it seemed to start flashing.
Okay.
I'm actually almost done, so
okay.
Just give me 2 minutes and then we got a session starting.
You didn't need
We were using it, but maybe we'll need it for the second session.
Okay.
Thank you. Okay, so once we have whatever we do to our FastQ, this is if we index it or we trim it or we remove bases from the end, we can then write out this FastQ2 file by using our write FastQ function, provide it the trimmed FastQ object, and then give it the file we want to write out to. Again, we can read in from FastQ, but we can also write to FastQ and write to FastQ.goes. So it will zip up the FastQ as we write. Something kind of hidden in our thing there. It's kind of interesting. I wonder what that secret word says. Can something do this all for me? Oh, interesting. So yes, there is something which can do this all for you, and this is the rFastP program. So rFastP was this kind of new, what it claims to be an ultra-fast, all-in-one FastQ preprocessor. So really what this is going to do is all these steps of trimming out reads which are low quality, removing the ends of reads which are low quality. So rFastP can do this in a really fast, efficient way. It isn't available from inside Bioconductor, but me and my colleague, Wei, and my team actually wrote up a little wrapper for rFastP. And this would allow you just to do very quick, it's got its own manual here, very quick quality control and plotting for your data. Okay. The nice thing about this is that it is significantly faster than this QA function. All we need to do is install the rFastP package. You can just do rFastP, give it the path to the data you want to process, tell it the path of the FastQ you want to create, the trimmed, tidied up FastQ, and it will go create this FastQ, and it will actually create an HTML report just like the QA function for you to review. By default, it's going to write that out to the report, but you can also actually take the output from the rFastP function, apply the QC function to this, and then it gives us a nice data frame summary, which I think sadly is falling off the end of the page. So reviewing a FastQ, reading it, trimming it is all fairly straightforward in R. The issue is that FastQs are actually like huge usually, and we have, as Illumina sequences have got bigger, FastQs aren't going to be handled in memory as well as they were when we only had 8 million units. Okay. So we need to take advantage of streaming these FastQs. We don't actually need the whole FastQ in memory. We only need to look really at a part of it, operate on that part, and then perhaps write that back out to file, continue with the next part, write out to file. Let me just try something quickly. So I am still teaching, but what's going to come up now is like a blue flashing screen. Okay. And it just didn't allow us to. So we have the FastQ sampler and the FastQ streamer functions, which allow us to kind of work through proportions of the FastQ file. So I think you can already see it's yellow. Yeah. Cool. So for evaluating the quality of our FastQ, we don't need the entire file. We just perhaps need to look at a certain number of the reads. Okay, so this is the idea that really, whatever, if your FastQ is poor quality, taking 100,000 reads from across the FastQ is as good as looking at every single read in the FastQ, right? Assuming it's randomly distributed, the error in the FastQ. The FastQ sampler is clever in that what it does, and here I've set it to be n equals 100, but what it does is it works through your entire FastQ file. And once as it's going through, it keeps a record of how many reads we actually want to extract. So here we're going to say n equals 100. And as soon as I've created this object, I can just say yield. And it's going to go and take 100 random reads from the FastQ. But what it's going to do really importantly is not just take the first 100 reads. And that's because actually FastQs are in order. FastQs are in order from the top left of the flow cell to the bottom right of the flow cell. So if I just took the first 100,000 reads, I'm taking the top left of the flow cell. There are positional effects on flow cells because actually you tend to add the liquid from the bottom. So the top may be affected differentially from the bottom. So by just taking the first 100,000, you may well just capture the top left and say your data's rubbish. And actually it got better towards the end. Okay, so FastQ sample is going to look across the whole flow cell and just pull us out 100 reads. Every time I want to take another 100 reads from it, I could do this again and it would give me out another 100 random reads. FastQ streamer is actually what we want to use if we want to work through the whole file from the top left of the flow cell to the bottom. So what FastQ streamer is going to do is allow us just to deal with a certain number of reads at a time. We will write them out to a file, and then we will deal with the next set of reads. Okay. So we set up the same way, but now we're going to do FastQ streamer. We'll give it the path to the FastQ file. We'll tell it how many reads we want to take out. So here, 100 in a go. And now if I press yield on my newly created FastQ streamer object, we're going to get the first 100 reads. If I do yield again, I will do the next 100 reads. Right? So I could do a loop here and just keep on taking data from my file until I'm finished processing the file. So what we're going to do in reality, what we would probably do is set this to 1,000,000. Here we're going to set it to 25,000. I've got my sampled FastQ object here. Right, so I'm going to create FastQ. And this is exactly how you would process this. Okay, you would do while, and you would say, in the while, we're going to do the yield. Okay, and while we still have reads to yield from this, so while when we yield, we still have reads of enough records that the length is greater than 0, it's just going to print how many reads we've yielded. So this is going to keep on going until when it runs the yield, it doesn't actually get any reads back. And that means the length of what it returns will be 0. It's not greater than 0, and it will just stop. So writing a while loop like this, operating on your FastQ within the while loop, writing it out to file, and then continuing for the next loop, the next loop, the next loop is how we can deal with really big files without having to worry about swamping our memory. So we've got some exercises now for the session, and they should be reasonably straightforward. We've got a little bit on how to write files, some of these quality controls to do. If I could recommend doing 2 plots, this plot is one of the most important plots. This is an essential FastQ quality plot, the drop of quality across time. And this plot is an essential plot. This is going to show you the biases or lack of biases for reads across time. Okay. I think what we're going to do, folks, is abandon this room for the rest of the day, because this isn't perhaps not going to get fixed. So is that okay with you two? Yeah, sorry. I'm also sorry, I found that kind of distracting. So I think I lost my thread a little bit in the middle. If you have any questions or I've missed something in the lectures, let me know. I can just tell you what we'll talk through now. Okay. I think it's distracting also to try and fix it. I should have not tried to fix it. I'll be back at 3, and I'll run through the exercises, but we'll do it on Zoom, if that's okay with everyone. For the next